sometime today, roughly, 100 billion of your cells will intentionally kill themselves. They will cleanly and methodically disassemble their own internal machinery, pack themselves into little digestible bubbles, and just disappear. And if it didn't do that, well, you would be alive to hear this deep dive.
It is a phenomenal, almost brutal, microscopic reality, right? We walk around feeling like one unified individual, a single entity with a single mind. But biologically, you are a walking, talking galaxy. You are a cooperative society of trillions of individual living factories.
Welcome to this deep dive. Settle in, because today we are going completely off the macroscopic map. We are exploring the ultimate building block of all life, the cell. And the mission today is, well, it's incredibly specific.
Yeah, we have our hands on an educational text that details the foundational biology of cells. But instead of just skimming it and giving you the bullet points, we are going to do something we rarely do.
Right, we're going to go line by line.
Exactly.
To make sure you walk away as a bona fide cellular expert, we are going to read through this source material step by step. We will present every single passage, read the exact text, and then we will completely unpack the biology, the history, and the physical mechanisms behind it.
Yeah, we are not just going to tell you what things are. We are going to tear them apart to understand how and why they work. No skipping ahead, no leaving you behind.
It's a literal microscopic journey. And understanding a cell isn't just about passing a biology test. It is about understanding how you, the person listening right now, function on an atomic level.
So let's jump right into section 1.
Yeah, the origins of life and the limits of sight. The document opens by taking us way back in time. Read the first passage here. Quote, "It is widely accepted amongst the scientific community that life originated in water. Some researchers believe that life may have originated in small water pools with changing environmental conditions rather than in the oceans. Hot springs are examples of such environments. In India, the hot springs of Puga Valley and Ladakh maintain very high temperatures, nearly at the boiling point of water, even in a cold climate. These environmental conditions seem to be similar to those on the early Earth, about 3.5 billion years ago. The organisms living in these hot springs are mostly heat-loving bacteria called thermophiles, which are unicellular," end quote.
Okay, let's unpack this.
Yeah, if we picture the dawn of life, we usually imagine like a calm, warm, primordial soup. But this text points to places like the Puga Valley and Ladakh, which is essentially a boiling, chaotic, volcanic kitchen.
Right.
Why would life start in an environment that actively destroys most living things today?
Well, what's fascinating here is the energy requirement for creating complexity. Life isn't a passive thing. To forge the very first organic molecules, to force atoms to bond together in new complicated ways, you need a massive amount of energy.
You need a spark.
Exactly. You need heat, you need chemical turbulence, and you need minerals. A boiling hot spring provides all of that. But it presents a massive problem. Heat destroys structure. The text introduces thermophiles, which translates from Greek thermo, meaning heat, and phile, meaning loving.
Heat-loving.
Right. These are unicellular organisms, single-celled beings that survive where they really should be boiled alive.
But how? I mean, if I drop a normal cell into boiling water, the proteins inside it literally unfold and cook, right? Like frying an egg. The clear part turns white because the heat destroys the folded shape of the proteins.
Spot on. So how does a thermophile survive? It's all about their microscopic architecture. The proteins in these heat-loving bacteria have evolved extra-chemical bonds, specifically tight ionic bonds and dense hydrophobic cores. They act like molecular superglue.
Oh, what?
Yeah, their proteins are physically lashed together so tightly that even at boiling temperatures, they just refuse to unfold.
That is incredible. They are born into the fire and chemically lock themselves together to survive it. But even before you get to complex proteins, life needed a basic shield.
Right.
The text continues with this exact point. Listen to this. Quote, "Scientists from the Birbal-Sahni Institute of Paleosciences, Lucknow, studied these hot springs and found that calcium carbonate formed rapidly around them. These deposits may have protected early organic molecules from harmful radiation and extreme conditions, and they may have also helped in the formation of the first protective membrane, the barrier that defines a cell." End quote. Wait, calcium carbonate? That's chalk.
Yeah, it's limestone.
That's wild.
It is. And if we connect this to the bigger picture, it answers one of the greatest mysteries of early biology. Before you can have a living cell, you have to have a boundary. If early organic molecules, the first fragile ancestors of DNA and proteins, were just floating in the open water, the intense ultraviolet radiation from the young sun would have shredded them instantly.
Because the early Earth had no ozone layer.
Precisely. They needed to hide. The scientists at the Birbalsani Institute are proposing that the rapid buildup of mineral chalk around these hot springs acted as nature's first hazmat suit.
Like little microscopic caves.
Yes. These microscopic pores in the rock provided a tiny shielded cup where fragile molecules could pool together, interact, and complexify without being blasted by radiation or washed away by boiling currents. Without this geological barrier, a biological membrane could never have evolved. It would just dissolve into the chaos.
It's like trying to build a house in a hurricane without putting up the external walls first. You need the scaffolding. Okay, the author then scales up from the very first cells to us. Let me read the next part. Quote, all living organisms are made up of cells. The cell represents the basic level at which life exists. Some organisms, such as bacteria or yeast, consist of only one cell, unicellular, while others like plants, fish, birds, or humans are made up of millions of cells, multicellular, that work together. A group of similar cells performing similar. Even when cells are organized into tissues, organs, and organ systems, the cell remains the fundamental unit of structure and function in all living organisms. This makes us wonder how such tiny cells perform so many different activities. What are the different components of a cell? How do cells in our body communicate with each other? Do cells live forever, or do they die? In this chapter, we will explore the answers to these questions as we enter the fascinating world of cells. End quote.
So we use words like tissues and organs all the time, but the jump from a single cell living on its own to a human being is a massive evolutionary leap.
It is the difference between a lone hermit in the woods and the entire infrastructure of New York City.
That's a great way to put it. The text is laying out the biological hierarchy. Think of a single cell like a brick. A unicellular organism like yeast is just one brick. It has to do everything itself. It has to hunt, eat, reproduce, defend.
Right.
But when organisms became multicellular, they discovered the power of specialization.
So a group of similar bricks gets mortared together to form a wall.
Yeah.
In our bodies, that wall is called a tissue, like muscle tissue or nervous tissue.
And you take 4 walls and make a room, which represents an organ like your stomach. Then you plumb a bunch of rooms together to make a house, which is an organ system like your digestive tract.
I see.
The beauty of this is that a single cell in your stomach doesn't have to worry about defending you from a virus or helping you see the color blue. It just focuses on secreting acid. It outsources the rest of its survival to the other cells.
But as the text emphasizes, no matter how big the house gets, the fundamental living unit is still that microscopic brick, which presents a massive logistical hurdle for us as humans trying to study them. How do we see the invisible? The text addresses this in the next section. Quote, 2.1 How to Study Cells. What do we call the ability of the human eye to see two very close objects as separate and distinct? Imagine 2 tiny dots drawn on a piece of paper. As the dots are moved closer, there comes a point at which they can no longer be seen as separate. When viewed from about 25 centimeters, the near point of human eye, 2 points separated by about 0.1 millimeter can be seen as distinct. Otherwise, they appear as a single point. This is called the limit of resolution of the human eye, which is 0.1 millimeter, end quote.
Okay, let's unpack this part.
Yeah, because I am struggling with the visual here. Why does our eye just, give up at 0.1 millimeters? Is it a focus issue?
It's actually not a focus issue at all. It's a physical hardware limitation. The back of your eye, the retina, is lined with photoreceptor cells, rods, and cones. Think of them like the pixels on a digital camera sensor. Each cone catches light and sends a single signal to the brain.
Oh, I see where this is going.
Right. If 2 tiny dots on a piece of paper are so incredibly close together that the light bouncing off both of them hits the exact same cone cell in your eye, your brain only gets one signal. It literally cannot know that there are 2 dots. It just registers one smudge.
Wow.
That 0.1 millimeter limit is the physical spacing of the pixels in your eye. If an object is smaller than that, or the gap between 2 objects is smaller than that, your biological hardware just tops out.
So if a cell is vastly smaller than 0.1 millimeters, peering harder or squinting does absolutely nothing.
Nothing at all.
We need a way to stretch the light before it hits our eye. And the text moves to exactly that solution. Quote, a cell is usually too small to be seen by the unaided eye. This raises an important question. How have cell biologists studied the structure and function of cells that are much smaller than the limit of resolution of the human eye? You have learned about convex lens. A convex lens, or a combination of lenses, i.e. an objective lens and an eyepiece, are used for the magnification of an object to make it appear larger, end quote. I know what a magnifying glass is. But on a physics level, how does a curved piece of glass solve the retina problem you just described?
It's all about bending light, which is called refraction. When light passes from air into glass, it slows down and changes direction. A convex lens is thicker in the middle and thinner at the edges.
Like a teardrop.
Sort of, yeah. When the light rays bouncing off a tiny invisible cell hit that curved glass, they are bent outward. They spread apart. By the time those light rays leave the glass and hit your eye, the image has been stretched out physically.
Okay, I'm following.
Now, the light from the left side of the cell hits one cone in your eye, and the light from the right side hits a completely different cone far away.
So we are essentially tricking our biological pixels by stretching the image across more of them. And that specific trick changed the course of human history. The text brings up the pioneer of this.
Yes, it does.
Quote, Robert Hooke was the first person to observe a cell in 1665 using a self-designed microscope capable of about 200 to 300x magnification. While examining a thin slice of cork, he observed small box-like compartments and named them cells. In school laboratories, light microscopes are used to observe objects using different objective lenses, for example 10x, 40x, to achieve better magnification and resolution under visible light. Explore different parts of a microscope in your school laboratory and use it to observe fine structures of various materials. Under the microscope, you'll see a magnified image of an object. Can you estimate its actual size? End quote.
Let's pause on Robert Hooke in 1665. The man was looking at a piece of dead wood cork with essentially two curved pieces of glass in a brass tube.
It's amazing what he achieved.
Hooke is a fascinating figure. He was a polymath, an architect, and a bitter rival of Isaac Newton. In 1665, he published a book called Micrographia, which was basically the first time anyone had ever shown the public what the microscopic world looked like. He had to draw everything by hand by looking through his primitive microscope.
But the funny thing is he didn't actually see a living cell, did he?
No, he really didn't. Cork is tree bark. It's dead tissue. What Hooke was actually looking at were the empty, rigid, dead walls of plant cells. All the living jelly inside had long since dried up and vanished.
So just the empty husks.
Right. He just saw these stark, empty, square grids. Because they looked like little bare rooms lined up in a row, he called them cells. He was referencing the small, Spartan, box-like compartments that monks lived in at a monastery.
That is wild. The foundational term for all of biology was coined because a guy looked at deadwood and thought it looked like a monastery.
It really is.
But as the text asks at the end there, once you are looking at something magnified 300 times, how do you actually measure it? You can't just stick a ruler into the lens, right?
Well, actually, that is exactly what the text suggests we do, but with a clever mathematical twist.
Wait, really? Let me read the experiment block then. This is activity 2.1. Quote, activity 2.1. Let us estimate the size of a cell 1. Take a transparent ruler with millimeter markings. 2. Place the ruler on the stage of microscope, focus on using the adjustment knob, and observe the diameter of the circular field of view through the eyepiece and measure it in millimeter. 3. Convert the diameter from millimeter to micrometer. Suppose the diameter of the visible field is 5 millimeters, meaning 1,000 equals 5,000 millimeter. 4. Remove the ruler and place an onion peel slide on the stage of the microscope. 5. Focus on the slide and count the number of cells present along the diameter of the field of view in one straight line. 6. Estimate the real size of the cell using the formula, unit conversion. 1 millimeter equals 1,000 micrometer. Estimated size of the onion peel cell. Diameter of the visible field in micrometer. Number of cells along the diameter. Suppose 25 cells are seen along the diameter. In that case, the size of one onion cell would be 5,000 millimeters. 25 equals 200 millimeters. End quote.
Okay, I know that's a lot of numbers.
Yeah, I have to be honest. Math in an audio format is incredibly tricky. I am completely lost. We are holding a ruler up to a microscope.
Let's visualize this together. Imagine looking through a submarine porthole. You want to know how big the fish swimming by are, but you can't go outside. First, you need to know exactly how wide your porthole window is. So you take a regular plastic ruler and hold it up right against the glass. You look through and you see that exactly 5 little millimeter tick marks fit across the circular window.
Okay, I'm with you. The window we are looking through is 5 millimeters wide.
Right. But a millimeter is a giant measurement in the cellular world. It's like measuring a car in miles, so we convert it to micrometers. There are 1,000 micrometers in a single millimeter. So our 5 millimeter window is actually 5,000 micrometers wide.
Got it. Thousands of tiny units.
Now, step 2. You take the ruler away. You take a thin slice of onion and slap it on the window. You look through the porthole and you see a row of onion cells lined up edge to edge, stretching from the far left of the window to the far right. You just count them. Let's say you count exactly 25 cells filling the width of the window.
Oh, so if the window is 5,000 micrometers wide and 25 cells perfectly fill that space.
You just divide the total space by the number of cells?
Right, 5,000 divided by 25 is 200. Every single onion cell is exactly 200 micrometers wide.
Yeah.
That is an incredibly elegant way to measure the invisible without needing a microscopic ruler.
It's the brilliance of deduction. And the text continues by explaining how the microscope itself manages to show us this view.
Let's read that. Quote, if the estimated size of an onion peel cell is 200 Bunner, how much does a light microscope magnify this cell? The total magnification of a microscope depends on its magnifying power of the eyepiece and the objective lens. If both the eyepiece and the objective lens have the magnifying power of 10x, then the total magnification will be 100x. This means that a cell with an estimated size of 200 nanometers will appear 100 times larger. Thus, a microscope allows us to see very small structures clearly and is an essential tool for studying the cell structure. Over the years, scientists have improved the microscope by improving its three main features: resolution, measure of clarity; contrast, the difference in brightness between various parts of an object; and magnification. These improvements have turned microscope into a powerful tool for studying cells. End quote.
So magnification is an additive.
Right. If I have a 10x lens near my eye and a 10x lens down near the slide, it doesn't just make it 20 times bigger, it multiplies the effect.
Exactly. The first lens makes the image 10 times larger, and the second lens takes that already enlarged image and makes it 10 times larger. 10 times 10 is 100.
Makes sense.
But notice the three features the author highlights: magnification, contrast, and resolution. Magnification is just making things bigger. But if you magnify a blurry image, you just get a giant, useless blur.
Right, which goes back to our pixel limit we talked about. That's where resolution comes in, the actual sharpness. And contrast is just being able to see a clear, translucent cell against a background of clear, translucent water. You have to stain it or alter the light to make it cast a shadow.
But eventually, light microscopes hit a hard physics wall.
Which brings us to the final part of Section 1. Let me read. Quote, apart from light microscopes, scientists also use powerful electron microscopes that reveal the fine details of a cell structure. These instruments use a beam of electrons instead of light to produce highly magnified images, allowing us to see cell structure at a nanometer scale with remarkable clarity. A nanometer is 1/1000th of a meter. You can see a picture of the lower surface of colocasia leaf observed under a scanning electron microscope. End quote.
Okay, this is a massive jump in technology.
Wait, let me challenge that. Why do we need to abandon light? Why can't we just build a bigger glass lens and keep magnifying the light endlessly?
Because light itself has a physical size.
Light has a size.
It is a mind-bending concept, but light travels in waves. The wavelength of visible light is roughly 400 to 700 nanometers long. If the object you're trying to look at, say a tiny virus or a small organelle inside a cell, is smaller than 400 nanometers, the light wave physically cannot bounce off of it.
What happens to it?
The wave just washes over it like an ocean swell washing over a tiny pebble. If the light doesn't bounce back to your eye, the object is invisible, no matter how powerful your glass lenses are.
That is staggering. The tool we use to see is literally too fat to touch the object.
Exactly. So in the 1930s, scientists realized they needed a thinner probe. Electrons act like waves too, but their wavelength is hundreds of thousands of times smaller than visible light.
That's tiny.
Very. By firing a beam of electrons in a vacuum chamber, bouncing them off a cell, and using magnets instead of glass lenses to focus them, we could suddenly see the nanometer scale, 1/1000th of a meter.
To put that in context, the jump from light microscopes to electron microscopes,
Mm-hmm.
it's the difference between looking at a city from an airplane and seeing the buildings versus zooming in from orbit with a military satellite to read the text on a discarded newspaper on the sidewalk. It completely changed the game.
It allowed us to see not just the walls of the cell, but the bustling, moving economy inside and outside of it.
Which is the perfect transition. Now that we have the ultimate camera, let's look at how the cell interacts with its environment. Let's look at the text for Section 2, the cell boundary, gatekeepers, and walls. I'll read the first part. Quote, 2.2 Structure of a cell. You have learnt that cells are organized into specialized tissues and organs and collectively perform specific function. For these cells to function as units, they must be able to interact with one another and with their surroundings. These interactions occur at the cell boundary, where substances move between the cells and their external environment. Even single-celled organisms exchange materials and respond to their environment through cell membrane. End quote.
This dismantles the idea of the cell as an isolated hermit.
Completely. A cell is a highly active chemical economy. It must take in raw materials, oxygen, glucose, amino acids, and it must export waste products like carbon dioxide.
Right, if it stops trading with its environment, it suffocates and dies in its own waste. The cell boundary is the border crossing where all this trade happens.
Let's read how this border crossing is managed. Quote, 2.2.1 Cell membrane. The universal feature of a cell. The cell membrane is a thin boundary that surrounds a cell and protects its contents. It defines the individuality of a cell and is also called the plasma membrane. The cell membrane is selectively permeable, which means it allows some substances to pass through it while blocking others. You have learned how oxygen and carbon dioxide move across the membranes of alveoli in the lungs. How does the structure of the cell membrane in the cells of alveoli control the movement of substances across it? End quote.
Selectively permeable is the heavy lifting phrase here. Permeable means things can pass through, like water through a sieve. Selectively means it chooses what gets a pass.
So it's not just a net.
No, I like to use the bouncer analogy. The cell membrane is like a highly trained bouncer at an exclusive nightclub. The bouncer lets the VIPs, tiny essential molecules like oxygen, stroll right in without even checking their ID. But if a massive, clunky molecule tries to get in or a toxic substance, the bouncer blocks the door. It is an actively managed border, not just a static wall.
And we can see this border in action with a brilliant, simple kitchen experiment the author provides. Let me read the activity. Quote, Activity 2.2. Let's experiment 1. With the help of a kitchen knife, carefully cut a potato into 2 pieces of roughly equal size. 2. Measure and record the initial weight of both the pieces using a weighing balance. 3. Put 1 piece of the potato in beaker A with plain water. 4. Put the other piece of the potato in beaker B with 20% salt or sugar solution. 5. Leave them undisturbed for about an hour, or until you notice a visible change in the size of the pieces. 6. Measure and record the final weight of each piece. 7. Calculate the difference between the initial and their final weights. What do you observe? You may observe that beaker A, the potato piece, swells. Beaker B, the potato piece, shrinks. End quote.
Okay, let's map this out.
Yeah, we have 2 identical potato chunks. One goes into plain tap water, the other goes into a heavily salted, dense brine. And after an hour, the one in plain water gets bloated and heavy, and the one in salt water gets shriveled up and light. Why? Salt doesn't, pull water, right?
You're right, salt doesn't have magical tractor beams. To understand this, we have to look at the next part of the text, which explains the invisible mechanics at play.
Okay, here's what the text says next. Quote, What do you infer? What do you expect in terms of changes in their weight? The weight of the potato piece in beaker A has increased, while the weight of the potato piece in beaker B is decreased. This happens because the cell membrane allows water to move in and out of the cell, but not the sugar or salt molecules. Water moves from an area with more water and less solute, dilute solution, to an area with less water and more solute, concentrated solution, until the concentrations in the 2 areas become equal. This movement of water through a selectively permeable membrane is called osmosis. End quote.
Okay, let's break down the kinetics of this. Osmosis, it sounds complicated, but it's really just molecules bumping into things, right?
Exactly. This is where we elevate the explanation. At a microscopic level, water molecules are constantly vibrating and bouncing around randomly. This is called Brownian motion. In the potato cell, you have water trapped inside a membrane. Outside the potato cell, in beaker B, you have the extremely salty water.
And the membrane, or bouncer, is selectively permeable. It allows tiny water molecules to squeeze through the pores, but the big, bulky salt molecules are totally blocked.
Right. So picture the pores in the membrane. On the inside of the cell, you have pure water molecules bouncing around. A lot of them randomly hit the pore and escape outwards. But on the outside, the water is crowded with massive salt molecules.
I see.
Because the salt is in the way, fewer water molecules successfully bump into the pore from the outside to enter the cell.
Oh, that makes perfect sense. It's just a probability game. Water is escaping out of the potato faster than it can get back in, simply because the salt outside is blocking the incoming traffic.
Yeah.
So the potato loses its internal water and shrinks. It shrivels up.
And in beaker A, with a plain water, the opposite happens. The inside of the potato naturally has some sugars and salts in it. The plain water outside has none. So pure water rushes into the potato without any obstacles, while the water inside the potato is blocked from escaping by its own internal sugars.
So the potato swells up.
Exactly. This natural striving for balance across a barrier is the definition of osmosis.
And this isn't just a fun potato trick. The text makes a massive connection to the real world here.
Yes, let me read that.
Quote,
You have learned in activities 7.8, dye spreading in water, and 7.9, fragrance spreading in air in grade 8, that particles of matter intermix due to a difference in their concentrations, also called a concentration gradient. Diffusion is the net movement of particles from a higher to a lower concentration, which occurs even without a membrane. Osmosis is the diffusion of water across a selectively permeable membrane. In plants, water from the soil enters root cells by this process of osmosis, end quote.
I love that. In plants, water from the soil enters root cells by this process. That is exactly how the massive 100-foot oak tree outside your window drinks water from the dirt.
It relies on the simple probability of osmosis.
Yeah. The root cells are saltier than the rain-soaked soil, so the water naturally forces its way into the roots to try and balance the concentration gradient. It's a completely passive, invisible pump that can lift water hundreds of feet into the air.
It is the silent engine of the entire botanical world, and the text provides the official scientific vocabulary for these states of balance, which are crucial for medicine and biology. Let me read the definition.
Go for it.
Quote, a cell is kept in salt or sugar solutions of different concentrations. Solute concentration of extracellular medium, solute concentration of intracellular medium isotonic solution, solute concentration of extracellular medium, solute concentration of intracellular medium hypotonic solution, solute concentration of extracellular medium, solute concentration of intracellular medium hypertonic solution, end quote.
Let's lock these terms in. Isotonic. Iso means equal.
Right. In an isotonic solution, the concentration of salt inside the cell matches the outside. Water moves back and forth evenly. The cell is perfectly happy.
This is why IV bags in a hospital are filled with a very specific, slightly salty saline solution. It has to be perfectly isotonic to your blood so your red blood cells don't explode or shrink.
Then we have hypotonic. Hypo means under or less, like hypothermia. There is less solute or salt outside the cell.
So water rushes in to dilute the cell and the cell swells up. If you inject pure hypotonic tap water into a patient's veins, their red blood cells will gorge on the water and physically burst.
Yikes. And finally, hypertonic. Hyper means over or more, like a hyperactive kid. There is way more salt outside the cell.
Just like our potato in the brine. Water rushes out of the cell to dilute the surrounding salt, and the cell violently shrivels and collapses. This is why drinking seawater when you are stranded on a raft will kill you faster than drinking nothing.
Because it pulls water out of you.
Exactly. The seawater is profoundly hypertonic to your body's cells. It literally sucks the water out of your tissues into your digestive tract, accelerating your dehydration.
That is terrifying and fascinating. So we know the membrane handles all this traffic, but what is this magical bouncer actually made of? The text gives us the structural blueprint. Quote, structurally, the cell membrane is extremely thin, about 7 to 10 nanometers thick. 1 nanometer equals 0.000000000000001 millimeters. The membrane has a lipid bilayer, 2 layers of special fat molecules with water attracting heads outwards and water repelling tails inwards, with proteins embedded in them. The molecules can move sideways, flip, and rotate within the membrane. End quote.
Fluid mosaic model and lipid bilayer. I know what fat is, but how does fat make a highly selective door?
This is one of the most elegant self-assembling structures in the universe. Let's look at the chemistry. These special fat molecules are called phospholipids. They have a split personality. They have a head made of phosphate that absolutely loves water. It's hydrophilic. But they have 2 long tails made of fatty acids that are terrified of water. They are hydrophobic. Oil and water don't mix, right?
Right, so if you drop them in the watery environment of a body, they must panic.
Exactly. The cell is full of water on the inside and surrounded by water on the outside. So the fat molecules spontaneously, without any energy required, arrange themselves into a double-layer sandwich.
Oh, I see.
The water-loving heads face outward toward the liquid, and the water-hating tails point inward, hiding against each other in the dry center of the sandwich. That is the lipid bilayer.
And because it's just fat molecules huddled together, it isn't solid. That's why it's called a fluid mosaic.
Yes. It's not a rigid wall of brick. It has the consistency of olive oil at room temperature. The protein molecules, our bouncers that are stuck into this fat layer, aren't cemented in place. They're bobbing around, sliding past each other, floating across the surface like buoys adrift in a sea of oil. It's a highly dynamic, liquid skin that holds you together.
But wait, if it's that liquid, how do things like trees stand up? A pine tree isn't made of liquid olive oil membranes. The text addresses this major difference between kingdoms of life.
Okay, let's read that part. Quote, neighboring cells through the cell membrane. However, cells of plant, fungi, and bacteria have an additional layer around the cell membrane called the cell wall. What do you think is the necessity of the cell wall in these cells? Activity 2.3. Let us investigate 1. Prepare temporary slides of a thin peel of an onion leaf or a Roeo cradle lily leaf and mount it with safranin using cover slip to observe plant cells under a microscope. 2. Similarly, prepare a temporary slide of cheek cells by gently scraping the inner side of your cheek with a cotton swab or the blunt end of a toothpick. 3. Spread the cheek cells on a clean glass slide. 4. Add a drop of water followed by a few drops of methylene blue stain and carefully place a cover slip. 5. Observe both the slides under a microscope. What do you observe? Onion peel cells or real leaf peel cells are box-shaped and regularly arranged, whereas cheek cells are irregularly arranged, figure point 8. Why do you think this difference exists? End quote.
So we are looking at the fundamental architectural difference between plants and animals here. Plant cells are boxy, they look like a brick wall. But human cheek cells are just irregular, sloppy, roundish blobs.
And we can actually stress test this difference to see why it matters. The author gives us a follow-up experiment. Let me read the stress test. Quote, prepare 2 slides of a Rio leaf peel and human cheek cells again, and put 20% sugar solution on them. Observe them under a microscope after half an hour. What do you observe? You must have observed that the boundaries of the plant cells remain the same, but their inner content shrinks, and the space between the inner and outer boundaries increases, figure 2.9a and b. You may observe that the cheek cells, on the other hand, have shrunk considerably, end quote.
Okay, so we are taking both the plant cell and the animal cell and dropping them into a 20% sugar solution, a heavily hypertonic environment.
Right.
We know from the potato that water is going to ruthlessly rush out of the cells to dilute the sugar.
Exactly. And the animal cheek cell behaves exactly like you'd expect. Because it only has that fluid olive oil membrane, it just completely shrivels up. It collapses like a deflating balloon.
But the plant cell is different.
Very different. The rigid outer boundary, the cell wall, is made of incredibly tough material. It stays perfectly intact like a solid cage. Only the living jelly inside the wall shrinks and pulls away from the edges.
It's like watching a tent collapse inside a sturdy wooden frame. The frame doesn't budge, but the fabric inside just falls apart. The text explains the evolutionary reason for this armor.
Let's read that quote. 2.2.2 cell wall. The outer covering of cells. In general, plants cannot move from place to place, so they need a rigid structure to withstand environmental stresses like wind and rain. Therefore, plant cells have an additional covering outside the cell membrane called the cell wall. The cell wall also helps leaves and flowers remain firm and maintain their shapes and help plants stay upright. Although rigid, the cell wall is permeable, which means water and some dissolved minerals can pass through it. Along with the selective permeability of the cell membrane, the permeability of the cell wall helps plant roots absorb water and nutrients from the soil. End quote.
If I'm an animal cell and I just watched my neighbor collapse in a sugar solution while the plant cell stayed structurally sound, I have to ask, aren't animal cells at a severe disadvantage? Why don't we have walls?
It's an evolutionary trade-off. Plants need walls because they're sessile. They can't run away. When a storm comes or a drought hits, they have to stand their ground. They don't have a skeleton made of calcium bone, so they rely on microscopic armor around every single cell to hold themselves up against gravity and weather.
But animals chose a different path.
Exactly. We chose movement. We need our cells to be squishy and fluid so our muscle fibers can contract and slide past each other. We need flexibility so our skin can stretch when we bend our knees, so we can run, hunt, and dance.
That makes sense.
If our cells were encased in rigid brick walls, we would be stiff as boards. We wouldn't be animals. We would just be fleshy trees.
The text summarizes that microscopic horror show we just talked about, the shrinking, with a specific term.
Quote, when we place a ruo leaf or an onion peel in a concentrated sugar solution, the plant cells lose water due to osmosis. However, the cells do not shrink in size because their rigid cell wall maintains their shape. The inner content of a cell shrinks as the cell membrane pulls away from the cell wall. This shows that the cell wall helps plant cells remain firm in their original shape. Animal cells, such as cheek cells, do not have a cell wall. Therefore, when placed in a concentrated sugar solution, they lose water and shrink. End quote.
The scientific term for the plant cell's insides tearing away from the wall is plasmolysis. It is a visual representation of severe dehydration in a plant. But what is this indestructible wall actually made of?
Let me read the final paragraph of this section. Quote, without a rigid cell wall, animal cells can change shape easily. This cellular flexibility supports the overall movement and functioning of animal tissues. The plant cell wall is primarily made of cellulose, a type of carbohydrate formed by many glucose units linked together. Cellulose in our diet acts as roughage, helping in digestion. Some microorganisms, like fungi and bacteria, also have a cell wall to provide protection and structural support to their cells. End quote.
Cellulose. We hear that word a lot in nutrition. It's a carbohydrate made of glucose. But glucose is just sugar. How does a plant build a brick wall out of sugar?
It's all about how the sugars are chained together. Think of Legos. If you stack them one way, they break apart easily. That's starch, which we can digest for energy. But plants link glucose molecules together using a very specific chemical bond called a beta-acetyl linkage. They weave these chains into long, ultra-tough microfibrils, sort of like spinning sugar into steel cables.
And human biology completely lacks the molecular scissors, the enzymes, to cut those cables apart.
Exactly. We literally cannot digest cellulose for energy. When you eat a celery stick, all that crunch you hear is your teeth physically shattering the cellulose cell walls. But chemically, your stomach can't touch it. It passes through your entire digestive tract completely intact.
Oh, that's why the text says it acts as roughage. It acts like a stiff biological broom, sweeping your intestines clean as it passes through.
So we have the dynamic fluid membrane, and we have the rigid cellulose wall. We understand the armor. But what are they actually protecting? Let's step inside. Let's move to section 3, the living factory. Inside the cell, here's the opening text. Quote, 2.3, the cell interior. A coordinated working system. You have learned that most cells have 3 basic parts. Y, a selectively permeable membrane called the plasma membrane. Y, a semi-fluid jelly-like substance called the cytoplasm. And Y, a prominent nucleus. In addition to the nucleus, the cytoplasm contains several subcellular components called organelles, along with other substances present in it, most of which are only visible with an electron microscope. Activity 2.4. Let us study 1. Study the given diagrams of a bacterial cell, a plant cell, and an animal cell. Figure 2.10a, b, and c. 2. Observe the different structures present in each of them. 3. Record your observations in Table 2.1. End quote.
We have breached the membrane. And it's not just empty space inside, it's filled with cytoplasm. When I hear jelly-like, I just picture an inert puddle of jello.
That is a very common misconception. Cytoplasm is not a quiet puddle. It is a wildly crowded, incredibly chaotic, hyperdense chemical mosh pit.
A mosh pit.
Yeah, it is packed with salts, proteins, and molecules constantly crashing into each other at millions of times a second. And floating in this intense fluid are the organelles.
Organelle literally translates to little organ. Just like my body has a heart and lungs to do specific jobs, the cell has little organs to do microscopic jobs. But not every cell is organized the same way, right? The text makes a crucial distinction here.
Let me read that quote. A bacterial cell lacks a well-defined nucleus and membrane-bound organelles surrounded by their own membranes. Such cells are called prokaryotic cells. Pro means primitive and karyon means nucleus. In prokaryotic cells, most cellular activities take place directly in the cytoplasm. In contrast, plant and animal cells have a well-defined nucleus and several membrane-bound organelles. Such cells are called eukaryotic cells. Eu means true, and karyon means nucleus. That's a quote.
Let's break down the Greek roots to make this stick. Pro means before or primitive, and eu means true or good. Karyon means kernel or nucleus.
Right. So prokaryotic means before the nucleus. These are bacteria. They are the ancient ancestors of life. I love the real estate analogy for this. A prokaryotic cell is like a really cheap studio apartment. There are no internal walls. Your bed, your stove, your toilet, your television are all just shoved into one open room.
Sounds messy.
It is. If you are cooking and doing laundry at the same time, the steam from the stove is getting all over your clean clothes. All the chemical reactions just happen together in the cytoplasm.
It's functional but chaotic. But eukaryotic, meaning true nucleus, these are the plants, the animals, and us.
Eukaryotes are the multi-million-dollar mansions. We evolved internal walls. We have a separate room sealed off for cooking, a separate room for the library, a separate room for waste disposal.
That's a huge advantage.
By putting membranes around our organelles, we can have highly toxic, highly acidic chemical reactions happening in one part of the cell without destroying the delicate DNA in another part. It allowed life to become infinitely more complex.
But if you have all these different rooms inside the mansion, how do things get from room to room? Do they just float randomly through the jelly? The text has the answer.
Let's read it. Quote, In eukaryotic cells, a network of fine fibers forms the cytoskeleton, which provides structural support, maintains cell shape, and enables cell movement and internal transport. It is visible only under an electron microscope as a separate entity. The cytoplasm may also store starch in plant cells or crystals of calcium oxalate or silica in some plant cells. These are known as cell inclusions, end quote.
Cytoskeleton. It provides structural support, which makes sense, but the text says it enables internal transport. How does a skeleton move things?
This is one of the most visually stunning mechanisms in biology. The cytoskeleton isn't just dead bone, it's an active highway system made of microtubules. When the cell needs to move a package of chemicals from one side to the other, it doesn't just throw it into the fluid and hope it floats there. It uses motor proteins like kinesin.
Kinesin. Wait, isn't that the molecule that literally walks?
Yes, if you look at an animation of it derived from electron microscopy, a kinase protein looks almost exactly like a tiny, weird, two-legged humanoid carrying a massive balloon over its head. It literally steps one foot over the other, trudging along the microtubule highway, burning energy with every step, hauling cargo to its destination.
It is a microscopic postal worker.
That is deeply unsettling and entirely awesome. So we have the building, we have the rooms, and we have the highway system. How do all the departments work together? The text gives us our guiding analogy.
Quote, cell organelles help in building new materials, removing waste, and providing energy to the cell. They work together to perform all functions of a cell. In other words, a cell is like a tiny living factory, with each of its parts doing a specific job. Let us explore these specialized structures present inside a cell, end quote.
Okay, a living factory. Every factory needs a central command. The boss's office, the text starts there, quote, nucleus house of coded instructions. The nucleus has a double-layered covering called the nuclear membrane, which has pores that allow the transfer of material between the nucleus and the cytoplasm. The nucleolus is the dense, round body in the nucleus where synthesis of ribosomal subunits take place. These subunits then exit the nucleus to cytoplasm, where one large and one small subunits assemble to form ribosome, end quote.
The nucleus isn't just sitting out in the open. It has a double-layered membrane. That is serious security.
It is the ultimate vault. The DNA inside is so precious it is the master blueprint of your entire existence that the cell spares no expense protecting it. The nuclear pores are like high-tech security doors. They don't just let anything in.
They check IDs.
They physically check the chemical ID of molecules, ensuring only authorized messengers can enter to read the blueprints. And deep inside that vault is a smaller, denser area called the nucleolus, which is churning out the parts for ribosomes. We will get to ribosomes in a second, but just know they are the factory workers.
But let's look at the actual contents of the vault. The text gets into the genetics.
Quote, the nucleus contains chromosomes, which are visible as rod-shaped structures only when the cell is about to divide. Chromosomes contain information for inheritance of characters from parents to the next generation in the form of DNA, the ribonucleic acid molecules. Chromosomes are composed of DNA and specific proteins. DNA molecules contain the genetic information. The functional segments of DNA are called genes. In a non-dividing cell, this DNA is present as part of chromatin material. Chromatin material is visible as an entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes, end quote.
Here's where it gets really interesting, because people use the words DNA, genes, chromatin, and chromosomes completely interchangeably, and they are not the same thing.
Let's clarify this step by step. DNA is the raw chemical code itself. Think of DNA as the alphabet, the letters A, C, T, and G. A gene is a specific sequence of those letters that forms a functional sentence. A sentence that says, make eye color blue, or build a digestive enzyme.
Okay, so the genes are the sentences made of DNA letters, but what is chromatin? The text calls it an entangled mass of thread.
If you took all the DNA in just one of your microscopic cells and stretched it out in a straight line, it would be over 2 meters long.
Wow.
That is an insane amount of data. To fit 2 meters of string into a microscopic nucleus, you have to spool it. Most of the time, the cell loosely wraps the DNA thread around proteins, leaving it a bit tangled but relaxed, so the cellular machinery can easily get in and read the sentences. That relaxed, messy state is called chromatin, like a bowl of spaghetti.
But when a cell wants to divide, a tangled mess of spaghetti is a nightmare to separate.
Exactly, you'd rip the DNA apart. So right before the cell divides, the chromatin tightly winds itself up into hyperdense, rigid, rod-shaped packages. It's like meticulously packing all your clothes into solid, movable suitcases before moving out of a house. Those tightly packed, transportable suitcases are the chromosomes. You only ever see the iconic X-shaped chromosomes when the cell is packing up to split.
It is an incredibly elegant filing system, but amazingly, the text points out that not every cell gets to keep this beautiful vault. Listen to this. Quote, some cells are specialized to perform specific functions. For example, mature red blood cells, RBCs, in humans do not have a nucleus, a nucleate. The absence of a nucleus provides more space for hemoglobin, allowing it to transport a larger amount of oxygen to all cells of the body. Since they lack a nucleus, they cannot repair or divide themselves. As a result, their lifespan is short, and they survive approximately for 120 days. End quote.
I want to linger on this because it is mind-blowing. A red blood cell is a living thing, but it intentionally ejects its own brain.
It is a profound, brutal evolutionary sacrifice. A red blood cell is essentially a delivery truck. Its only job is to haul oxygen. Mammalian evolution eventually realized that the massive nucleus was taking up too much valuable cargo space in the truck. So as the cell matures in your bone marrow, it literally spits its own nucleus out.
It just chucks it.
Yeah, it throws away its DNA, its blueprints, its entire command center, just to pack in a few more molecules of hemoglobin to carry oxygen.
But because it threw away its instruction manual, it's a ghost ship. It can't build new proteins, it can't fix a leak. If it gets damaged, it's doomed.
Right. It has a strict, inevitable expiration date of about 120 days. It circulates in your blood until it gets battered, fragile, and old, and then your spleen filters it out and destroys it. It sacrifices its own immortality for the greater good of keeping the rest of the body highly oxygenated.
Moving on from the boss's office, we head down to the actual manufacturing floor of the factory. The text reads, quote, Endoplasmic reticulum, ER. Manufacturing factory. The endoplasmic reticulum, ER, is a large organelle that spreads like a network within the cytoplasm of the cell. The ER is continuous with the outer membrane of nuclear envelope. The ER plays a key role in the synthesis and transport of proteins, fats, lipids, and some hormones in some of the specialized cell. The structure of the ER in a cell varies depending on its function. There are two types of ER. Y-rough endoplasmic reticulum, RER. It looks rough under an electron microscope because it has ribosomes attached to its surface and is mainly involved in protein synthesis and protein secretion, for example, in gland cells such as pancreatic cells. Y-smooth endoplasmic reticulum. It does not have ribosomes on its surface and therefore looks smooth. It is involved in the synthesis and storage of fats and hormones. End quote.
The endoplasmic reticulum, it's physically attached to the nucleus, like a massive factory complex branching right out of the command center, and it's split into two divisions, rough and smooth.
Let's start with the rough ER. The text says it looks rough because of ribosomes. Remember the nucleolus inside the vault making ribosome parts? Those ribosomes exit the nucleus and stick directly to the folds of the rough ER. Under an electron microscope, they look like thousands of tiny rough studs, like sandpaper.
And what are the ribosomes actually doing?
They are the 3D printers of the cell. They read the genetic code from the nucleus, and they physically link amino acids together one by one to print custom proteins. Since the rough ER is covered in them, it is the dedicated protein assembly line.
And the smooth ER, no ribosomes, so no protein printing.
Right. It looks like a network of smooth, branching tubes. Its job is completely different. It manufactures fast lipids, which the cell needs to constantly rebuild that fluid mosaic membrane. It also makes steroid hormones like testosterone or estrogen. Plus, in your liver, the smooth ER acts as a detox center, breaking down drugs and alcohol.
Okay, so the factory has built the raw products. We have proteins from the rough side, fats from the smooth side. Now what? You can't just dump them into the cytoplasm. The text explains the next step. Quote, Golgi apparatus, the packaging and shipping centers. If you carefully look at the diagram of a cell, figure 2.10b and c, you will notice stacks of flattened, sac-like structures. Together, these stacks form the Golgi apparatus. It is functionally linked to the ER, the cell membrane, and the other cell organelles. The Golgi apparatus acts like the cell's post office. It modifies, sorts, and packages proteins and or lipids into vesicles for transport, secretion, or lysosome formation. Meet a scientist. The Golgi apparatus was first observed in 1898 by an Italian scientist, Camillo Golgi, in the nerve cells of a barn owl. Using special staining techniques, he observed a thread-like network. Early microscopes could not resolve it clearly, therefore, many doubted its existence. However, electron microscope observations confirmed it decades later. End quote.
The Golgi apparatus is the Amazon fulfillment center of the cell.
It's the perfect analogy. The ER builds the raw, unfinished proteins and drops them off at the Golgi. The Golgi is a stack of flattened pancakes. As the protein moves through each layer of the pancake, it gets modified. Maybe a sugar molecule is added as an address label. Finally, the Golgi pinches off a piece of its own membrane, wraps the protein in a tiny shipping box called a vesicle, and fires it off to its final destination.
I want to talk about Camillo Golgi for a second. The text gives us this Meet a Scientist block, 1898. He is staring at the nerve cells of a barn owl, and he creates this thing called the black reaction, a silver stain that suddenly makes this weird, ghostly network of pancakes visible. But no one believes him.
It was incredibly controversial. The scientific community literally thought he was seeing an optical illusion, or that his silver stain was just coagulating and creating fake structures. They called it an artifact. He spent decades defending his discovery. He even won a Nobel Prize, and people still doubted him. It wasn't until the 1950s, when the electron microscope was invented, that scientists could zoom in and say, oh my God, Golgi was right all along. It's really there.
The ultimate delayed vindication. Okay, so the factory builds things and it ships things. But any heavy manufacturing process produces garbage. Machinery breaks. How does the cell handle waste? The text tells us, quote, lysosomes, the cleanup system cells, produce waste materials and damaged, worn-out organelles during their activities. How does the cell prevent these wastes from accumulating inside it? Lysosomes are single membrane-bound sacs filled with enzymes, which can break down unwanted proteins, carbohydrates, fats, and even crusts. Human sperm cells contain lysosomal enzymes. When a sperm meets an egg, these enzymes help break down the outer layer of the egg, allowing fertilization to take place, end quote.
Lysosomes are the recycling centers, or the garbage disposals. But earlier I asked you how they work. You said they use enzymes. What is an enzyme actually doing to break down waste?
An enzyme is a highly specialized protein that acts like a pair of molecular scissors. The lysosome is essentially a tiny bubble filled with a violently acidic cocktail of these hydrolytic enzymes. Let's say a mitochondria gets old and stops working. The cell can't just leave it there to rot. The lysosome floats over, completely engulfs the dead mitochondria, and unleashes those enzymes.
And then what?
The enzymes physically sever the chemical bonds holding the old organelle together, chopping it down into basic reusable raw materials. It literally digests it.
But wait, if a lysosome is full of acid and scissors that dissolve organelles, why doesn't it dissolve itself? Or what happens if it leaks?
That's the genius of it. The membrane of the lysosome is specially coated to resist its own acid. But if the cell is severely damaged or dying, all the lysosomes will deliberately pop at once, flooding the cell with acid and dissolving the entire cell from the inside out. They're sometimes called suicide bags for this reason.
And here is a mind-blowing macroscopic connection. The text says human sperm cells use lysosomal enzymes.
Yes. The tip of a human sperm cell has a specialized giant lysosome called an acrosome. When a sperm is trying to penetrate a human egg, it doesn't drill through mechanically. It detonates that lysosome, spilling the corrosive garbage disposal enzymes onto the thick outer shell of the egg, physically melting a hole through it so the sperm's DNA can enter.
If we connect this to the bigger picture, this microscopic organelle designed for waste disposal was repurposed by evolution to be the fundamental key for the creation of human life. The scale of that is just beautiful.
So we have the boss, the assembly line, the shipping center, the garbage disposal. But to run a factory, you have to pay the electric bill. You need power.
Let's read about that. Quote, mitochondria. The powerhouse of the cell mitochondria are often called the powerhouses of the cell because they supply the energy needed for most cellular activities. Each mitochondrion is surrounded by two membranes, HUI. The outer membrane is smooth and porous, phi. The inner membrane is folded into finger-like projections called cristae, which increase the surface area for chemical reactions and facilitate energy production. In mitochondria, the glucose and other molecules are broken down to release energy during a process called cellular respiration. You have already studied about it. The energy released is stored in the form of a molecule called adenosine triphosphate, ATP, which acts as the energy currency and is used for most of the cellular activities. End quote.
The powerhouse of the cell. It is the most famous phrase in all of biology. Everyone remembers it from high school. But let's look closer. It has two membranes, which is unusual, and the inner one is folded up into these things called cristae. Why the origami?
It's a brilliant engineering solution to a space problem. The actual physical machinery that generates power is embedded directly on the surface of that inner membrane. If the inner membrane was just a smooth balloon, you'd only have a limited amount of surface area, which means limited power generators.
So the folding helps.
Yes. By aggressively folding the membrane back and forth into cristae, you can cram a massive amount of surface area into a microscopic space. More surface area equals more space for chemical reactions.
And the result of those reactions is ATP, adenosine triphosphate. The text calls it the energy currency.
Think of ATP like a rechargeable battery. When the mitochondria burns glucose from the food you eat, it uses that energy to jam a third phosphate group onto a molecule, locking it in place like compressing a powerful spring. That's ATP. It floats out into the cell. Whenever a motor protein needs to take a step, or the bouncer at the membrane needs to forcefully eject a toxin, it grabs an ATP, breaks off that third phosphate, releasing the compressed spring, and uses that burst of energy to do work.
And then it gets recharged.
Exactly. The dead battery goes back to the mitochondria to be recharged.
Every single heartbeat, every thought in your brain is powered by billions of these invisible springs snapping open. That is how animals get energy. We have to eat glucose. But plants found a cheat code. The text moves to them next. Quote, plastids center for food synthesis in the plant cells and beyond. You have learned that mitochondria provide energy to the cell that comes from food. Animals can obtain food from their surroundings, however. Plants synthesize food in the presence of sunlight. But where do plants synthesize their food and obtain energy for cellular activities? Plants use special organelles called plastids for food synthesis and storage. End quote.
Plastids are a category of organelles that are entirely unique to the botanical world. Animals just straight up don't have them.
We missed out on a great evolutionary trick. Let me read the next part about the most famous plastid. Quote, as you have learned, plants prepare their food by the process of photosynthesis in the presence of sunlight. A green pigment called chlorophyll, which is present in the chloroplast, a type of plastid, absorbs sunlight. Chloroplasts are double membrane-bound organelles, like mitochondria. Inside the chloroplast, there is a semi-fluid substance called the stroma. Within the stroma are disc-shaped membrane structures that contain chlorophyll. Light energy is absorbed. End quote.
The chloroplast, it contains chlorophyll, which makes plants green. But what is it actually doing with the light?
It is engaging in quantum biology. The chloroplast acts like a microscopic solar panel. When a photon of sunlight travels 93 million miles through space and strikes a chlorophyll molecule, it excites an electron, knocking it out of orbit. The chloroplast harnesses the kinetic energy of that rogue electron and uses it to violently force carbon dioxide and water molecules to bond together, creating glucose. It is literally sorting solid food out of thin air and sunshine. That singular organelle is the foundation of almost all food chains on planet Earth.
But not all plants are green. The text asks a great question here. Quote, are there any other plastids in plant cells that contain any pigments other than the green pigments? How do flowers, fruits, and vegetables acquire varied colors? In flower petals and fruits, plastids contain pigments other than chlorophyll. These plastids are called chromoplasts. In Greek, chroma means color. Their pigments, maybe yellow, orange, or red, are the source of bright colors in such flowers and fruits. Such bright colors help in attracting pollinators for pollination and fruit-eating animals that help in seed dispersal. End quote.
Chromoplasts. Chroma means color. So when an apple turns from green to bright red or a flower blooms brilliant yellow, it's because these specific plastids are producing different pigments.
And it is a genius evolutionary strategy. Plants can't walk around to find mates or spread their children. They have to bribe animals to do it for them. The vibrant red of a chromoplast in a berry is a literal billboard advertising to a bird, hey, come eat this, it's full of sugar. The bird eats the berry, flies 10 miles away, and poops out the seed in a new location, complete with a little bit of fertilizer. The chromoplast is the marketing department of the plant.
And there's a third type of plastid which does no marketing at all. Let me quote. Some plastids lack pigments and are thus colorless. These are called leucoplasts. In Greek, leukos means white. Leucoplasts store food material, such as starch, oils, or proteins, and are classified based on the type of food they store. For example, some leucoplasts in potato and taro colocasia cells store starch. End quote.
Leucoplasts. Leukos means white or clear. Since they are underground in a root, they don't see sunlight and they don't need to attract animals.
So they become invisible storage bins. They are the plant's deep pantry. When the leaves up top make too much sugar during the summer, they convert it to starch and send it down to the roots to be packed tightly into leucoplasts. That's why a potato is so heavy and dense. It is a massive cellular warehouse.
Okay, one more major organelle to cover in our factory tour, and this one solves a physics problem for plants. The text reads, quote, Vacuoles, the organelles for storage and support plastids, such as chloroplasts, help plants produce food and temporarily store it. Other plastids, such as leucoplasts, help in storing food. But where are water, minerals, and waste materials stored in the cell? Why do plants look wilted when they do not get enough water? End quote.
We talked earlier about the rigid cellulose cell wall holding the plant up. But a wooden frame isn't enough on its own. Let's read how the plant manages this. Quote, in a mature plant cell, there is usually one large central vacuole surrounded by a single selectively permeable membrane. The vacuole is filled with a watery fluid called cell sap. The vacuole stores water, minerals, sugars, and waste material. By storing large amounts of water, the vacuole helps maintain pressure inside the cell, which keeps a plant cell firm. When a plant does not get enough water, the vacuole loses water, the cells become less firm, and the plant gets wilted. In animal cells, vacuoles are sometimes present. Although they are not as large as plant vacuoles, they help in the temporary storage of materials. End quote.
In a plant cell, the vacuole isn't just a tiny storage closet. It is often the largest organelle in the cell, sometimes taking up 90% of the internal volume. It is a giant internal water tower.
And it relies on a concept called turgor pressure. Because a plant doesn't have a skeleton made of calcium bones, it relies on hydraulics to stand upright. The central vacuole fills with water, blows up like a massive balloon, and pushes outward with immense force against the rigid cellulose cell wall. That intense internal pressure keeps the stem rigid and the leaves flat and broad to catch sunlight.
So when you forget to water your houseplant, the roots run dry. The vacuole slowly loses its water to osmosis, the balloon deflates, the internal hydraulic pressure drops to zero, and the heavy cell wall sags under gravity. The plant physically wilts.
Animal cells don't need to do this. We have literal bones and muscle tone for structure. So our vacuoles, if we have them at all, are tiny temporary storage bubbles, not giant structural water towers.
The final part of this section details an experiment that borders on science fiction. It brings everything we've learned about the organelles and the nucleus together. I'll read the text. Quote, next, they took another closely related bacterium and removed its DNA, but kept the rest of the cell, such as the cytoplasm and the cell membrane, intact. They inserted the synthetic DNA into this cell. After this, the cell started to grow and divide, following the instructions from the newly inserted synthetic DNA. This experiment showed that DNA controls the structure and activities of a cell. However, scientists did not create a completely new cell from scratch. Only the DNA was synthetic. The other parts of the cell were taken from an already existing living cell. End quote.
Wait, hold on. Scientists built synthetic DNA in a lab. They wrote a chemical code.
Yes. This is referencing groundbreaking work from researchers like Craig Venter. They essentially booted up a computer, typed out a sequence of A, C, T, and G to code for a very basic bacterial genome, and then chemically printed that raw DNA string in a lab.
And then they surgically scooped out the natural DNA of a living bacteria, leaving the empty shell, the ribosomes, the membrane, the cytoplasm. They injected their printed artificial DNA into the empty shell, and the cell rebooted.
It woke up, and it started following the human-written instructions. It perfectly illustrates the philosophical division of the cell. The DNA is the software. The rest of the cell, the membrane, the ribosomes, the mitochondria, is just hardware. Hardware is dead, useless metal without an operating system. But if you swap out the software, the exact same hardware will run a completely different program.
It is the ultimate, undeniable proof that the nucleus and its DNA are the absolute master controllers of the living factory. It really makes you wonder what life actually is if we could just hot swap the code like a video game cartridge.
Yeah, it's pretty incredible.
So we know how the factory is built, we know how it runs, and we know who is in charge. But how does this hardware multiply? How do we get from a single fertilized egg to a walking, talking human being? Let's move to Section 4, Growth, Division, and the Unifying Theory.
The text begins with a very practical question. Quote, 2.4, how do normal cells grow and divide? When you get a small cut on your skin, it heals after a few days. When hair fall out, new hair grow back. How does this happen? It happens because cells in our body can grow and divide to replace the old, dead, or damaged cells. When our body grows, it is not just because cells get bigger. Cells can grow only up to a certain size, but growth happens because cells divide to form new cells. Let us study freshly growing root tips of an onion to understand how cells divide. End quote.
This clears up a massive misconception. When a baby grows into an adult, it is not because their individual cells are inflating like water balloons.
Right. An adult's skin cell is roughly the exact same size as a baby's skin cell. You are bigger simply because you have more of them.
But why? Why can't a cell just keep growing until it's the size of a basketball?
It's dictated by the square cube law in physics. As a cell gets bigger, its internal volume, the cytoplasm, increases much faster than its surface area, the membrane. The cell relies on its membrane to bring in food and push out toxic waste. If the cell gets too big, the internal volume demands so much food and produces so much waste that the tiny surface area of the membrane physically cannot keep up with the traffic.
That would choke on itself.
Exactly. The cell would poison itself or starve. So before it reaches that critical breaking point, it hits a size limit, copies its DNA, and divides perfectly in half to reset the ratio.
And we can see this division happening in real time in a lab. The text gives us another experiment. Quote, Activity 2.5. Let us enhance our skills. 1. Take a jar and fill it up with plain water. 2. Now place an onion bulb over the jar in such a way that its base-bearing roots is immersed in the water. 3. Leave this setup for 5-6 days and observe. Do you observe the roots growing? Cut 2 to 3 centimeters of the freshly grown roots and transfer them to freshly prepared acetoalcohol, glacial acetic acid, ethanol, 1.3. Keep the root tips in acetoalcohol for 24 hours, and then transfer them to 70% ethanol for preservation. 4. Take 1 or 2 preserved roots, wash them in water, and then place them on a clean slide. 5. Put 1 drop of dilute hydrochloric acid, HCl, on the root tips to soften the tissue. Rinse the roots after 10-15 minutes, then add 2-3 drops of acetocarmine stain on them. 6. Leave the slide for 5-10 minutes and then gently warm it with caution over a spirit lamp. 7. Cut the tip portion of the root on the slide and put a cover slip. Gently squash the cover slip with your thumb to spread the cells on the slide. End quote.
This is an incredibly detailed lab procedure. Acid, ethanol, staining, warming over a lamp. Why go through all this trouble just to look at an onion?
Because catching a cell in the absolute precise moment of division is like catching lightning in a bottle. You need a tissue that is growing violently fast. The very tip of an onion root is actively pushing and grinding through the soil, constantly creating new cells to grow longer. So the odds of catching cells in the act of splitting are high there.
And what's the acid for?
Remember that incredibly tough cellulose cell wall we talked about? You have to dissolve that armor. You soak it in hydrochloric acid to melt the cell walls into mush. Then you add the acetocarmine stain, which chemically binds specifically to DNA, turning it bright red. Finally, you smash the tissue flat with your thumb so you only have a single layer of cells.
That sounds brutal for the cell.
It is, but if you look under a microscope, you won't just see a nucleus. You will literally see the bright red chromosomes, those packed suitcases, being pulled apart to opposite ends of the cell right before it splits down the middle.
It's the mechanics of life laid bare. And this frantic division isn't just happening in onions. The author scales this up to us. Quote, every day, an estimated hundreds of billions of cells in our body are replaced, which is almost 1% of the total number of cells in our body. Both prokaryotic and eukaryotic cells divide, but eukaryotic cells divide in a more controlled and orderly manner by a process called the cell cycle. You will learn about the stages of cell division in higher grades, end quote.
Hundreds of billions of cells replaced every single day.
The math is staggering.
So what does this all mean for us? It means you are constantly regenerating. You are not a static, solid object. You are a flowing river of cellular matter. Roughly 1% of you is brand new every single day. The harsh acid of your stomach destroys its own lining so fast that you have a completely new stomach every few days. The outer layer of your skin is entirely replaced every month.
It's like the ship of Theseus.
Exactly.
Mm.
You are essentially the ship of Theseus, constantly swapping out the planks but maintaining the shape.
Let's read about the specific mechanisms that allow those planks to be swapped perfectly.
The text reads, quote, 2.4.1 Cell division. Cell division is the process by which new cells are formed from pre-existing cells. It allows living organisms to grow, repair damaged tissues, and reproduce. Some cells, such as skin cells, divide continuously to replace cells that are lost regularly. There are 2 major types of cell division: mitosis and meiosis. Mitosis is important for normal growth, repair, maintenance, and asexual reproduction, while meiosis is important for sexual reproduction for creation of genetic diversity. End quote.
2 paths, mitosis and meiosis. Let's look at the first one, which handles the everyday repairs.
Quote, mitosis: every human begins life as a single fertilized egg. This one cell divides repeatedly to form trillions of cells in the body. Cells increase in number through mitosis, which is the most common type of cell division. Mitosis produces 2 genetically identical daughter cells from one parent cell. Each new cell gets the same DNA and the same number of chromosomes as the parent cell. This ensures that genetic information is largely maintained across body cells. End quote.
Mitosis is all about making perfect clones.
Exactly. When you scrape your knee on the sidewalk, your skin cells don't want to get creative. They don't want to experiment with new genetics to heal the cut. They just want to make exact 100% identical copies of the skin cells that were violently scraped away to plug the hole perfectly. The parent cell meticulously duplicates all of its DNA, splits right down the middle, and you are left with two identical daughter cells.
And scientists have learned how to harness this cloning ability outside the body. Quote, scientists have developed methods to grow plant and animal cells outside the body in special conditions. This is called cell culture. In this process, cells are taken from an organism and placed in a nutrient-rich medium that allows them to grow and multiply. To keep the culture safe, the right temperature, acidic or alkaline conditions, and moisture under sterile conditions are maintained. Cell culture is crucial for studying how cells work and for the production of biochemicals, food, medicines, vaccines, and more. End quote.
Cell culture. Taking human cells and putting them in a petri dish, feeding them a pink nutrient broth and letting them just endlessly undergo mitosis.
It is the absolute foundation of modern medicine. By tricking cells into undergoing mitosis in a controlled lab, we can do miracles. If someone has horrific burns, we can take a tiny stamp of their healthy skin, put it in a culture, and let mitosis grow sheets and sheets of custom, perfectly genetically matched skin to graft back onto their body. We cultivate massive vats of bacterial cells engineered to produce human insulin for diabetics. We test the safety of highly toxic new drugs on a plate of cultured liver cells to see if it kills them without having to risk the life of a human patient.
It is incredibly powerful. But this power to divide endlessly has a very dark side if the cell loses control. The text addresses this head-on. Quote, the processes of mitosis and meiosis must occur in a proper and controlled manner. What happens if meiosis and mitosis do not happen properly? If there is any error in these processes, it can lead to various problems in the body of an organism. Errors in mitosis lead to uncontrolled cell divisions. This can lead to the formation of tumors and abnormal number of chromosomes in body cells. Errors in meiosis may result in genetic disorders, which may be associated with developmental problems or distinctive physical features. Faulty meiosis may also cause early pregnancy loss or reduced fertility, end quote.
If we synthesize this, cancer is fundamentally a disease of mitosis.
Yes. Usually a cell's DNA has strict checkpoints. There are genes, like the famous p53 gene, known as the guardian of the genome, whose entire job is to slam on the brakes and say, stop dividing now, the cut is healed, we have enough cells.
But what if it breaks?
Well, if a mutation caused by UV radiation, or chemicals in cigarette smoke, or just a random copying error damages that specific brake pedal in the DNA code, the cell goes rogue. It just undergoes mitosis endlessly, creating clones of itself over and over again, piling up into a tumor. It's a cellular factory that refuses to shut down its assembly line, even as it crushes the surrounding tissues.
And what about errors in the other type of division, meiosis? The text poses a really interesting thought experiment here, referencing an Indian scientist. Let me read it. Quote, Arun Kumar Sharma was a famous Indian scientist known for his work on chromosomes. As a botanist, he is known for his work in the area of plant taxonomy, evolution, and development. He also invented many useful lab methods to study chromosomes in plants. For his contributions to botany, he received many honors, including the Shanti Swarp Bhatnagar Award and Padma Bhushan. 7. If the skin cells start dividing by meiosis instead of mitosis, what do you think will happen to a cut on the skin? End quote.
Okay, to answer this thought experiment, we have to understand what meiosis actually does. We know mitosis makes a perfect clone. What is meiosis doing differently?
Unlike mitosis, which preserves the exact genetic code, meiosis is the specialized division that creates sex, cells, sperm, and eggs. Its entire purpose is to intentionally shuffle the DNA deck and cut the number of chromosomes strictly in half. It takes genes from your mother, genes from your father, recombines them into totally unique patterns, and prepares them to merge with another human to create a genetically distinct offspring. It creates diversity.
So applying that to the thought experiment, imagine you cut your arm. If your body mistakenly used meiosis to heal it, the cells wouldn't make exact clones. The new patch of skin healing the cut would scramble the DNA to make the new cells.
It would be biological chaos. The new patch of skin healing the cut would have completely randomized different genetics from the rest of your arm. It might be a different color, it might sprout different hair, it might have completely different texture. You would heal as a patchwork chimera. It wouldn't be you anymore. That's why your body strictly segregates meiosis to reproduction and uses mitosis-boring, reliable clones for repair.
All of this knowledge, the microscope, the organelles, the DNA, the division, culminates in one grand unifying idea. The text lays out the history, quote, 2.5 cell theory. the unifying principle of biology. An important observation about living organisms is that all organisms are made up of cells. In 1838, a German botanist named Matthias Fleiden reported that all plants are made up of cells. In 1839, German zoologist Theodor Schwann found that all animals are also made up of cells. Later, in 1855, a German scientist named Rudolf Virchow further expanded the cell theory by stating that new cells are formed only from pre-existing cells. Together, their work led to the formulation of the cell theory. According to the classical cell theory, all living organisms are made up of one or more cells. Why the cell is the basic unit of structure and function in living beings. Why all cells arise from pre-existing cells. This unifies all biology, from bacteria to humans, and explains life's continuity through cell division." End quote.
This is the E = mc squared of biology.
It truly is. Schleiden looked at leaves and stems. Schwann looked at muscle and bone. They realized that despite the macro differences, the foundational brick was identical. It brought the plant and animal kingdoms under one unifying rule, and Virchow added the final, most profound piece. Cells don't magically pop into existence from mud or dead matter. Spontaneous generation is a myth. A cell can only come from a previous cell dividing.
Which means, and this gives me chills, there is an unbroken physical chain of cell division stretching from the living cells inside your heart right now, going back through your parents, through your primate ancestors, through the early mammals, the fish, the single-celled eukaryotes, all the way back 3.5 billion years to those boiling hot springs in the Puga Valley. You are the continuous, unbroken tip of a 3.5 billion-year-old dividing lineage.
It is the ultimate continuity of life. But if they keep dividing forever, how do they know when to stop in a localized area?
Let's read the next part. Quote, 2.5.1. Do cells grow and reproduce forever? Cells grow and divide in a controlled way, stay in the right place, carry out their functions, and eventually die when they are no longer needed. Dead cells are replaced by new cells that carry out the same function. Thus, every cell has a definite lifespan. If cells do not die when they should, or if they die too early, problems can arise in the body. In many animal cells, cell division usually stops when cells come in contact with neighboring cells. This process is called contact inhibition. However, cancer cells lose this control and keep dividing uncontrollably, leading to the formation of tumors. Plant cells grow differently. Due to their rigid cell walls, plant cells do not show contact inhibition and follow a different pattern of growth. End quote.
Contact inhibition. That sounds like a brilliant biological sensor. How does a cell know it's touching a neighbor?
The membrane isn't just a barrier, it's covered in touch receptors, specifically proteins called integrins and cadherins. When an animal cell divides to fill an empty space like a cut, these proteins are floating freely. But the moment the cell physically bumps into another cell on all sides, these cadherin proteins lock together like a molecular handshake. That physical connection sends a chemical signal straight to the nucleus that says, "We're crowded, hit the brakes, stop mitosis."
And as we mentioned, cancer cells are deaf to that signal. They have mutated receptors. They bump into their neighbors, but the signal never reaches the boss's office, so they just keep pushing and dividing anyway.
The text takes a moment here to just step back and marvel at all this coordination.
Let me read this interlude. Quote, "Even though cells are extremely tiny, they perform remarkable functions. They produce energy, synthesize and secrete substances, divide to form new cells, and work together to maintain the proper functioning of the body. Every living being, from a tiny bacterium to a giant tree, is made up of cells. You are one of the products of these tiny, hardworking cells." End quote.
It is truly awe-inspiring, but sometimes that coordination requires absolute sacrifice. The text gets into the hook we open the show with.
Quote, "How do cells monitor their growth to maintain a balance? Cells also have natural ways of dying to maintain a balance. Programmed cell death, PCD, is a genetically regulated and organized process of selective cell destruction. It is essential for normal development, cellular quality control, and immune function. When an embryo develops, the PCD helps form fingers by eliminating cells between digits. Without it, we would have webbed hands." End quote.
Programmed cell death, also known as apoptosis. We often think of death as a failure of biology, an accident or an infection. But at the cellular level, death is a required tool for sculpting life.
The webbed fingers analogy is perfect. When you were an embryo in the womb, your hand didn't start as 5 separate stalks growing outward. It looked like a solid, flat, fleshy paddle. Your genes didn't build the fingers up. Instead, the genes told the strips of cells between the fingers to commit mass suicide.
And it isn't a messy death, right? If they just exploded, it would cause massive inflammation.
Exactly. Apoptosis is incredibly clean. The cell activates specific enzymes called caspases. These act like internal demolition crews. They systematically chop up the DNA, neatly package the organelles into little membrane-bound, shrink-wrapped bubbles, and then the cell signals the immune system's macrophages to come quietly eat the bubbles. They cleanly disassemble themselves, carving out the empty space so your fingers could be separate. The empty space is just as important as the structure. Again, when this fails, disaster strikes. The text reiterates, quote, "How do cancer cells grow and spread? Normal cells grow, age, and die in a controlled manner. Sometimes this system breaks down and abnormal cells start growing and dividing uncontrollably. This results in the formation of tumors, which may be benign or malignant. Cancer's tumors can invade nearby tissues and even spread to other parts of the body to form new tumors." End quote. Cancer ignores the stop dividing signal of contact inhibition, but it also ignores the kill yourself signal of programmed cell death. It refuses to die when it's damaged.
It becomes functionally immortal. And the final sentence there, spread to other parts of the body, is metastasis. The cancer cells mutate further, breaking those coherent handshapes that anchor them in place, and they float away in the bloodstream to seed new rogue factories everywhere. It is biology's most tragic rebellion.
The final piece of text from our source brings up a historical scientific proposal that hints at the sheer potential of the cell. Quote, "In 1902, the Austrian botanist Gottlieb Haberlandt proposed that any," end quote.
And that's where the specific sentence in the source trails into the summary block. But the concept Haberlandt proposed, which the author is referencing, is totipotency.
Totipotency.
It is the idea that a single mature plant cell has the total potential, all the genetic information required, to regenerate an entire fully formed plant all by itself. It proves that even when a cell specializes to become a leaf or a root, it never throws away the master blueprint. Every single cell retains the full code of the whole organism. It's the basis for cloning.
To wrap up our incredibly deep dive, let's do a rapid-fire recap of the summary points the text provides to lock in everything we've learned. Here's the first point, quote,
Organisms. Why prokaryotic cells do not have a well-defined nucleus. Instead, their genetic material is present in a region called the nucleoid. They also lack membrane-bound organelles, end quote.
Remember the chaotic, boiling hot springs? The cheap studio apartment? No internal walls, no boss's secure vault.
Quote, why eukaryotic cells are larger and more complex. They have a well-defined nucleus and several membrane-bound organelles, end quote.
The multi-million-dollar mansion. Specialized rooms for specialized jobs, protecting the DNA in a vault.
Quote, all cells are surrounded by a cell membrane. In addition, cells of plants, fungi, and bacteria have a cell wall outside the cell membrane, end quote.
Every cell has the fluid mosaic bouncer at the club door handling osmosis. But plants and bacteria also have a rigid brick wall of cellulose outside the club to stand up against gravity. Quote, why? The nucleus of eukaryotic cells contains chromosomes, which are composed of DNA and associated proteins, which carry genetic information. End quote.
The boss's office holds the DNA sentences, the genes, which pack up into dense chromosome suitcases when it's time to divide.
Quote, why? All cells are filled with cytoplasm. In eukaryotic cells, cytoplasm contains several cell organelles, each performing a specific function. End quote.
The chaotic, crowded jelly fluid holding the microscopic factory equipment.
Quote, why important cell organelles include the nucleus, endoplasmic reticulum, mitochondria, Golgi apparatus, ribosomes, and lysosomes. End quote.
The boss, the smooth and rough assembly lines, the folded power plant, the post office, the 3D printer workers, and the acidic garbage disposal.
Quote, why plant cells also contain special organelles called plastids, such as chloroplasts, leucoplasts, and chromoplasts. End quote.
The quantum solar panels, the hidden starchy pantries, and the colorful marketing billboards for pollinators.
Quote, we mitosis produces two daughter cells identical to the parent cell. End quote.
Making perfect clones to heal your scraped knee and grow your body.
Quote, my meiosis is a two-step division process which produces 4 daughter cells, each having half the number of chromosomes. End quote.
Shuffling the genetic deck to make sperm and eggs, ensuring no two humans are exactly alike.
Quote, mammal cells grow in a controlled manner, perform their functions, and die naturally, while cancer cells lose control and keep dividing uncontrollably, leading to the formation of tumors. End quote.
The beautiful, fragile balance of life relies entirely on knowing when to stop dividing and having the grace to die when necessary.
Wow. If you are listening, congratulations. You have just survived a comprehensive step-by-step masterclass on the absolute foundation of biology. You've walked through the selectively permeable gates of the membrane, watched motor proteins march down the cytoskeleton highway, and stared into the DNA vault.
Before we go, I want to leave you with one final provocative thought. We discussed two very distinct cutting-edge concepts today. We talked about programmed cell death, where cells have a built-in biochemical code telling them to cleanly dismantle themselves. And we talked about that wild experiment where scientists inserted printed synthetic DNA to completely rewrite a bacteria's operating system.
Right.
If we connect those two things, if we can already write synthetic software to run a cellular hardware, and we know there are natural pre-existing codes for apoptosis or cell death, how close are we to simply coding human cells like we code computers?
Yeah.
Could we, in the near future, write a biological software update injected into a patient and literally broadcast a master signal that tells every single rogue cancer cell to execute its own programmed death sequence and simply turn itself off?
That is profound. It turns biology from a mystery into a programming language. It's something for you to ponder as you think about the trillions of tiny, hardworking factories operating perfectly inside you right now. Remember, the next time you look in the mirror, you aren't just seeing one thing. You're looking at a vast, cooperative galaxy of billions.